So the selected abstracts will be presented by Alexandros Sofias. He recently moved to New York to join the Mulder Lab. We're very happy with that. He's a PhD student at the Norwegian University of Science and Technology at Trondheim. And today he'll present some beautiful results of research he has done there. And his presentation is titled Hitchhiking with Immune Cells, Dominant Tumor Targeting Mechanism for RGD Modified Nanoparticles. Good luck.
Perfect. Okay, hello everybody. My name is Alexandros. As Mandy mentioned, I'm a PhD candidate at the Norwegian University of Science and Technology. Would like to share with you my research. So my research focuses on understanding the in vivo mechanism of action of nanoparticles and also study their interactions with immune cells. And as I will show you today, those specific interactions with immune cells are very important in order to deliver nanoparticles to the tumor. So in addition, I would like to introduce you also with the idea of hitchhiking. So nanoparticles hitchhiking with immune cells. And then I would like to start my presentation with some introduction for people that are not so familiar with the nanoparticle world. So a nanoparticle utilized in oncology has two possible ways to reach the tumor. One is passive accumulation, and the alternative is active targeting, so targeting towards the receptor. In this idea, we have nanoparticles which we decorate with ligands, for example, antibodies or peptides that have really good affinity for a specific receptor on a cell. So unfortunately, even though this idea with active targeting is like very cool and could be promising for several types of tumor targeting, we don't have in literature a lot of quantitative data in terms of showing the exact mechanism that those nanoparticles work in vivo. And this is what we try to do in this specific project, to understand how those nanoparticles work. So in order to develop active targeting nanoparticles, we had to select a specific ligand. So in this project, we selected the RGD peptide. So this peptide is widely used. There are a lot of studies that focus on conjugate this RGD peptide on nanoparticles. As you can see also from the plot, year by year we have more and more manuscripts on that topic. One more reason why we selected to make RGD nanoparticle is that those nanoparticles, this RGD peptide target one specific integrin, so the alpha V beta 3 integrin, which is overexpressed in the angiogenic vasculature. So those endothelial cells that surround tumor express this integrin, and this integrin can be really accurately targeted by the RGD peptide, as we believe. So, and this is what I want to point out. So the current opinion on the field is that RGD nanoparticles, after they are being injected in the circulation, they target directly those integrins that are overexpressed in the endothelial cells. And this is one of the most representative images in literature. You can see on the left RGD nanoparticles and right control nanoparticles. And you maybe can see those like small speckled pattern, and authors often claim that this is active targeting of RGD nanoparticles toward the integrin receptor. But this opinion, we are challenging it, and we believe that this is not the case, and this is not how those nanoparticles work. So for that, we wanted to build a solid strategy in order to prove exactly how RGD nanoparticles work in vivo. What we did is we have a mouse that we inoculated with 41 breast tumor cells, and after a couple of weeks, the tumors were big enough in order to visualize them with intravital microscopy. And you can see the whole process, how we cut the skin and how we flap it, and we can create this small window chamber and then visualize the tumor. And our mouse model is cool in terms of visualization because the GFP, the endothelial cells express the green fluorescent protein, so we can really accurately and nicely visualize the lineage of the vessels. And also by administering different compounds, we can also visualize cells that are extravasated, immune cells that are extravasated in the tumor. In addition, because we can produce good quality data, we are able to image in depth, and then we can have also some nice Z-stack. You can see, for example, here this small hole in the endothelium, how nicely can visualized in Z direction. So we modified our nanoparticles with this RGD peptide, and for first time we injected them in vivo. So what's happening? What we saw in the previous video, yeah, maybe we can see it also a bit here, is that yeah, we see nanoparticles in red that have suddenly targeted vessel walls, so they have suddenly targeted the endothelium. And then what we did is repeating this experiment with fast imaging. We co-injected RGD nanoparticles in red and control nanoparticles in green. So we wanted to visualize real time how nanoparticles behave in vivo. And we saw this really unexpected thing, that RGD nanoparticles are taken up by cells, and we can see cellular movements around the vessels. So both nanoparticles actually taken up, but RGD nanoparticles are taken up in higher extent. So we wanted to further investigate this, and first of all, we want to clarify and understand that those big red spots we observed are indeed cells. So what we did is injected intravitally CD45 monoclonal antibody in green, and you can see that the antibody goes directly and stains those big red spots, which means that this is like the validation that those spots is not something random, but they are cells which have taken up RGD nanoparticles. And then we were able actually to visualize further movements which are RGD nanoparticles that have taken up by immune cells, and then the immune cells extravasate, and we can actually visualize them in the tumor environment. So what was interesting in that, we wanted to follow this pattern, and initially we have the GFP expressing endothelium. So usually the endothelium has a nice continuation, so you see like a clear green line. But in that occasion, after we have injected the RGD nanoparticles, we see some black holes interrupting the continuation. And then we presumably believe that those were immune cells. And by following visualizing the process, we saw another cell which enters from the circulation and penetrates within the endothelial cells. So then by merging with a nanoparticle channel that you can see in red, we see that this effect is not something spontaneous, but are nanoparticles taken up by cells, and the cell penetrate within the endothelial cells junctions. Further, we now are start to be convinced that a hitchhiking process occurs. So nanoparticles intravitally injected in the circulation are taken up by immune cells, and those immune cells then leave the circulation and enter into the tumor environment. So for understanding which cells are responsible for taking up RGD nanoparticles, we performed a detailed flow cytometry experiment in which we isolated blood from mice injected with RGD nanoparticles or control nanoparticles, and we limited our options into immune cells, so either neutrophils or Ly6C low monocytes, which are also, those are the tumor-associated macrophages. And one very important detail in this slide that you can see is that neutrophils are taking up RGD nanoparticles in higher extent in comparison to the control nanoparticle. But this is not the case about macrophages, which take up equally all nanoparticles. So then we repeated flow cytometry in tumors. So we injected again a mouse either with RGD or control nanoparticles, and we isolated the solid tumor, and we did single cell suspensions, treating with antibodies. And in this slide you can see two very important details. First of all is that neutrophils indeed take up RGD nanoparticles in higher extent than control nanoparticles. And second is that in the endothelial cells, you can see in the fourth figure, that we don't have any significance in favor, in uptake in favor of one of two nanoparticles. This is one more verification that the initial idea that nanoparticles target directly the endothelial cells is not correct. So we are now almost sure that the way that RGD decorated nanoparticles enter the tumor environment is by hitchhiking using neutrophils. And then we see a nice pattern in which neutrophils in blood take up RGD nanoparticles at earlier time point, and then they progressively deliver it after 24 hours to the tumor. So to summarize, we have a new suggested mechanism which says that RGD decorated nanoparticles do not target the integrin receptor directly, but what is happening is that nanoparticles first are taken up by immune cells in specific neutrophils, and then the neutrophils leave the circulation and localize in the tumor microenvironment. So last but not least, we have repeated all those experiments by using the three most widely used type of nanoparticles, so nanoemulsions, lipos, and micelles, and we have the exact same behavior. So we saw a universal applicable knowledge now on how RGD nanoparticles behave and how we can utilize them in order to reach and target tumor. So that was my presentation, and of course I would like to acknowledge first of all my supervisor, Surt Hak, assistant professor at NTNU in Trondheim, Norway. You can see him in action while scanning mice. And Christine and Niels, technicians, helping us setting up our mice experiments, and Niels is responsible for breeding those GFP expressing endothelium mice. And of course, for this year, 2018, I will be here at Mount Sinai, work for Professor Mulder. And I would like to thank, first of all, him and Professor Ochando for their valuable help so far in my research, and all members of Mulder Lab for the collaboration we have. And they make me feel very welcome here, so thank you.
Thank you for a beautiful talk. We have time for a couple questions.
Super nice work, very elegant. I imagine you have had some pushbacks, but your work reminds me, I mean, the concept you are suggesting have been shown in other diseases like multiple sclerosis, but not in an active way, with iron oxide particles that were taken by monocyte and crossing the blood-brain barrier. Basically the same thing, hitchhiking, but I imagine that the decoration of the particle are going to be driving which type of cell are going to be hitchhiked. So can you comment on that if you were to target the brain versus... And the other question I have, you would rely on inflammation. So if you have no inflammation, you can hitchhike. Right?
Okay, so to answer the first, so what we want to build ideally over time is a library in which we identify how different ligands are used in order to target specific cell subsets. In specific, I didn't mention that for sake of time, but RGD and nanoparticles target specifically neutrophils, because in neutrophils we have co-expression of alpha V and beta 3 integrin. So this is not well characterized, because in a lot of nice studies showing this so-called active targeting towards the receptor, and all the uptake by nanoparticles, they consider it as side effect. So we have seen that integrins are expressed in neutrophils, and that's why we can actively target them. So of course, hitchhiking, now we study different types of tumors. So we have 41 tumors, and we have 2 different derivatives. Those derivatives, one is metastatic, the other is not, and they have different constitution in immune cells. So one tumor is really poor in neutrophils, the other is very rich in neutrophils. And now we are in process of understanding if that's like super important, the presence of this specific immune cell to harness this hitchhiking effect. But this is, of course, I know that there are other studies showing hitchhiking via immune cells, but for RGD nanoparticles, to the best of my knowledge, hasn't been described before.
I'm sorry, we're kind of out of time, so I'm gonna give you this one.
Thank you very much.
We have a 10-minute break, please. If you want to ask questions, we'll be available. But let me take a picture.
There's time for a photo.
They're only being nice to you now, but you'll wait later.
Okay.
Okay, thank you
Thank
for
you.
that. Yeah.